Okay, we're going to start off with looking at DNA replication a very simplified way, and then we're going to bring in all the different components and the important requirements for DNA replication. What is all the types of additional molecules that we require to replicate this molecule, okay? So initially it says here, DNA replication is semi-conservative, and you'll notice there was a question similar to that on your last set of homework, okay? So that means that each of the two strands in a double-stranded DNA molecule act as a template to produce two new strands, okay? So one of the first things we need for DNA replication is a template strand, i.e. we need the molecule because we need to take a copy of this molecule, okay? So again, very simplified before we look at it in more detail. So four different sections, okay? So here is our DNA, our double helix, okay? So essentially what happens is we talk about it being in a helix, we talk about it in a strand. So what happens is the first thing is it has to unwind, okay? So it looks literally like this, okay? And what's happening here is we have our two open strands. It's no longer in a spiral. And as you can see, we've got our nucleotides. They're complementary. A is always with T, G is always with C. These little dots represent the hydrogen bonds, okay? So essentially, if I take you back to this diagram actually, okay, when we're talking about here, so these are anti-parallel strands. Here, we class these as weak hydrogen bonds because this eventually, as we're just about to talk, has to open up, okay? However, when we talk about this sugar phosphate backbone, these also are joined, so the individual nucleotides are joined together via hydrogen bonds also, but they're stronger bonds, okay, because they are not needing to break away or open up from each other. It's just these bonds here, all right? So back to this diagram. So essentially, we have unwound our spiral, our helix, and we now have our two open strands, okay? And again, we can see it's anti-parallel. There's a 5 end, and there's a 3 end, and then the opposite strand is running in the opposite direction, 3 end to 5 end, okay? So these hydrogen bonds, so the second part is, these hydrogen bonds are going to break, okay? And this opens up the two strands to make it wider, okay? Because essentially what we're wanting to do is we want these strands opened up so we can have free DNA nucleotides, just the individual nucleotide with one base, to come along and bind on to its complementary partners, okay? So on to number 3. So it says each strand of DNA acts as a template for synthesis of a new complementary strand. So I've tried to keep it all the exact same. So here is this strand here, as it says, old strand, and then this strand here, the old strand, is exact same as this, exact same as this. But what's happened is it's opened up now, okay? And then these individual nucleotides have came along and binded onto its complementary partner, i.e. this is an individual nucleotide. So this is guanine, that's bound onto cytosine. So individual nucleotide, cytosine, and that's binding on to guanine, and so on and so forth, till it creates this new strand, okay? And you'll see that this new strand, okay, is synthesized 5 to 3 direction. Again, we'll talk about that in greater detail in the next video. And it's the exact same. So what's happening is we have two strands, but we are also creating two new strands at the same time, because this is going to be our new molecule, our new DNA molecule, okay? So again, here is thymine, and then this nucleotide adenine is going to come and bind onto that. Guanine is going to bind onto cytosine, thymine to adenine, and so on and so forth. And again, the new strand has been synthesized from the 5 end to the 3 end, okay? And then finally, as it says here, replication produces two identical DNA double helixes, each one with one new and one old strand. Okay, so here is our new strand, and here is our old strand, and so on and so forth. Okay, so here's our two new molecules.